So, welcome to this month's ReproNim webinar. I guess I get to introduce myself. Given the folks who are here, I don't know that too much introduction is necessary, but this is a video that will be available. So I'm Dave Kennedy. I am nominally the principal investigator of the ReproNim initiative, leading the cats of the program that brings you ReproNim. In our webinar series, we've spent, many times talking about our own technology developments. We've spent a number of our sessions looking at other people's technologies and also looking at applications. At least once a year, I like to take one of these meetings and use some sort of high level, state of the ReproNim type of presentation. And it's particularly apropos this year, this time of year, as we are approaching our phase 3 renewal application in the upcoming months. So without further ado, I will slog through the state of the ReproNim. So we're going to talk about the past, the present, and the future. And again, as I foreshadowed, the competing renewal for this project is going to be submitted May 27th-ish. So thank you all for listening and helping me to organize my thoughts around that topic. While it's my pleasure to get to represent the group today, all of this work, of course, is carried out by lots of individuals, many of whose pictures I could find on the web, but you see the brains for to represent the folks who I didn't happen to find quickly the slides on the web. But anyway, not only is it lots of lovely individuals who are working on this, it is a number of esteemed institutions that make this possible. You see the things here, and so no further ado on that. I couldn't find a nice thing for Nina's company, so that's just Nina. I will introduce a little bit about the background of ReproNim. To some extent, all of you attending today, are well steeped in ReproNim lore, but it's good for us to practice this and also good for others to see it in the video version of this. We'll go through some historical developments about tools and some current work that's going on in the success stories about those tools, and then spend the last little bit talking about the future of where we're going and our interactions with the community. So I should start by reminding ourselves that, the ReproNim is a NIBIB funded P41, what used to be called Biomedical Technology Research Centers. It was initially funded in April of 2016. Its highest level objective is to enhance the reproducibility of the way neuroimaging research is performed and reported in the neuroimaging literature in order to extend the value of our national investment in neuroimaging research while making the process easier and more efficient for investigators. That objective is summarized in our vision statement, which would be, all neuroimaging is reproducible neuroimaging, and the detailed delivery mission is to deliver effective tools, training, and principles to the community to support the entire workflow from acquisition to publication in terms of rigorous, reproducible, and fair neuroimaging techniques and practices. And as always, interrupt at any point in time. I don't really see questions or chat or people, so make yourself known if you'd like to interrupt or discuss any topic, of course, along the way. The premise to ReproNim since its inception was that the neuroimaging field is actually pretty well endowed with resources to make reproducible neuroimaging a reality. We have well-established standards for data. We have a stable and nice set of established tools that are in common use, and thanks to NIH and other fundings, there are a good number of repositories that are available to facilitate data sharing and resource sharing. So these resources ought to facilitate the complete transparency of the execution of neuroimaging research. But take a look at the literature, take a look at the state of the art. Adoption of all of these lovely practices in terms of using them in reproducible fashions remains the exception rather than rules. Yeah, most of the literature you read does not have the data accessible. Most of the literature you read does not have the complete description of the processing and sharing of the complete set of results in a way that results can be confirmed or denied. Our conjecture, therefore, is not that the neuroimaging literature cannot be transparent, but rather that it is not transparent. Why, you might ask? Well, the answer, some folks will tell you, because it's still too hard. It's too time consuming. It's not valued enough. The value proposition for research, does not, reward doing the efforts that are necessary to embody reproducible neuroimaging research. Rather, the grants and publications and career tracks, are dominated by purely publication counts, not assessment of how right or persistent or reproducible those observations are. So again, that's the battle that we started facing in the original version of this, and the battle we continue to face as we go forward. P41s nowadays, have a 15-year horizon. As I said, we started in 2016. Our phase one was resource development. I already started with the fact that a lot of core resources, tools, standards, and repositories existed, but there was still a good bit of missing technology in terms of the use and additional technology needed. So our phase one was really a resource technology development phase. Phase two, the phase that we're currently coming towards the end of, is refinement of these resources, trying to get towards adoption. Whereas phase three, the future that we're going to apply for shortly, will really begin to concentrate further on the adoption side of the puzzle. And this evolution is driven by the concerns, of course, that technology shouldn't exist for technology's sake. We need to be developing technologies, yes, but we need to be developing technologies that actually find adoption in the community and actually lead to improvements in the behavior of the community using the resources and procedures that are adopted. I put these little pie charts in that kind of, from a effort point of view, sort of, show that, the development, was kind of, high in the beginning and, has to, by definition, decrease a little bit as we, begin to, focus more and more on refinement and adoption of these techniques so that we, guarantee to our reviewers and to the funders, that we're developing technology that is useful for the community. By way of introductory types of things, I'd like to reiterate some of the principles and the evolution of some of our principles that guide the work that we're up to. We started back in the early days of ReproNim with a guide called the 5 Steps to More Reproducible Neuroimaging. Those were nice and concise, but tricky and broken up into lots of sub-steps. So that was a good guideline. But more recently, that has evolved into some reproducible neuroimaging principles and actions, which are a little more concrete and a little more tailored to the actual resources and things that investigators can do. And finally, I'm going to spend a moment on something we called YODA, which is Yoda's organogram on data analysis, which again is sort of a higher level standard, a higher level principle that we sometimes forget to keep an eye on when we try to put together our processes in the most reproducible, most comprehensive, most self-describing ways possible. And again, to set the stage, I want to remind ourselves of the kind of quintessential neuroimaging research workflow, which starts on the left with some sort of data acquisition. That can be new imaging, new subject experiments, or data acquired from public and or repositories of various sorts. You start from your raw data, you apply some sort of analysis, you derive some sort of results, and perform statistics to come up with conclusions and inferences, and then you publish that, and hopefully you distribute and share the contents and the details of those publications in a publicly accessible with the appropriate access restrictions as publicly accessible as possible. So that's kind of the stages that we're concerned with. Everyone in the neuroimaging community again needs to solve each of these uh, boxes in one way or another. And again, people do this and have been doing this since the beginning of time. It's just that they're done in fairly ad hoc ways. And how can we help to decrease the ad hocicity and increase the reliability and comprehensiveness of the procedures that we have to do to get through this workflow. Our original, 5-step, kind of followed, this high-level workflow. It looked at issues, that underlie the reproducibility of study design. It looked at issues that related to data collection, looked at issues related to data processing, looked at issues related to statistical analysis, and looked at issues related to publication. And this generated, a large number of, actionable things, but they're a little bit, sort of across the board. But the organizing principle here was that going from current what one does often to complete reproducibility is very, very hard, and there's lots of steps to it. So the point here was can we break things up into smaller steps, into bite-sized pieces that are more likely bite offable one thing at a time. So the goal is not to get people to complete reproducibility, but to incrementally increase the skills and the abilities of folks to operate in a re-executable fashion. So to take sort of 5 high-level things and break them up into smaller bite-sized pieces, so that a given individual could do one at a time, and each time they add one, they're doing a better job of overall reproducibility. And that was fine, and that guided some things along the way. More recently, in thinking about how we promote the actual resources that we have, and how we think about the details of the workflow, we've kind of reorganized those original 5 steps into 4 sort of high-level principles that wrap themselves around study planning, data and metadata management, software management, and publication. And each of these principles can relate to a set of core actions. And those are the use of standards, the application of annotation and provenance monitoring, the use of version control, and the use of containers. So every principle that we consider, it can be represented in a number of actions that help make that principle operate. So this is kind of the new pivot to the original 5 steps. Now becomes these ReproNim principles and actions. I want to take a minute to remind ourselves, of YODA. Again, it's a concept that's been around for a while, but again, it's kind of a higher level principle that I, for one, don't always remember as much. And again, I think it's important for us to remember this. And thanks to Austin for summarizing this, and I stole it from him, thanks to being in a public repository. So I wanted to go through these to again help us all, sort of keep these ideas in mind. The YODA ideals, include the fact that YODA compliant data sets, contained the portable computational environments to compute the results within that data set. And within that data set, YODA compliant data sets preserve the provenance of the computational procedures that generated the derived data. And YODA compliant data sets strive to embody reproducibility within the data set itself. This is associated with principles that say the essential computational information must be included in the data set. The elements of the data set must be version controlled. The elements may include sub data sets, or directly include the data set itself. The provenance of all modifications to the elements of the data set must be annotated, and the data structure should accommodate standards, and all assets should be organized in a modular structure. Now, those are kind of nice general words, but again, I think it's useful to look at that in the context of this kind of diagram. Again, Michael and Yarrick and the Data Lab team have promoted this kind of idea for a while, but again, finally I'm sort of embracing it a little bit more to present that here. Now, we're all used to BIDS, the brain imaging data structure, which helps us organize our imaging MRI imaging studies nicely, so that it's much more self-described. So it really lowers the barrier to a sharer in having to explain anything about the data to a sharee, to someone using the data, because all the metadata is there, and everything's named in a standard fashion, and the standards for the organization are well understood and well followed. A BIDS data set is very self-describing. So that's great for the data set itself. But in general, in our research world, we don't always publish data sets, we publish research studies. And that research study also needs a framework, a container in which the entire research study can be shared and self-describing in a way that both supports the executability and minimizes the effort to explain what a data set is to others through the use of standards. So in addition to the imaging data that might be nicely in BIDS, a YODA compliant data structure will also include the containers that were used upon the data that you're publishing, linked together so that we don't have to separate these, that they're together. And the YODA compliant container includes the results of the operation of this container on that data to generate some particular result. Well, that should all be kept together, and then that result is used in some particular analysis. That should be kept together, and all of that goes into some particular paper. And so that whole track from the raw data, that raw data's source, perhaps from OpenNeuro, the container, the derived result, and the analysis, the complete package of materials that went into said paper should all be kept together so that can be shared and in a sort of holistic and sort of comprehensive fashion. Right now, again, it is relatively easy to share each of these different pieces in different places. You might have your BIDS data accessible at OpenNeuro. Your container might be at Docker Hub. Your derived data might be out some S3 bucket. Your analysis might be in some Python notebook somewhere, and your paper could point to all of those. But keeping those together in the sort of YODA compliant, complete, sort of descriptive, way, so that data that stays together is more likely to be, stay together as opposed to have link rot and things like that. So again, we spend a lot of time talking about, oh, we should containerize things, or oh, you should share, data, or oh, we should make sure that there's version control on all of our processing and our results. But the higher level is that all of these things should be kept together to be comprehensively describe the study that gets published in your particular paper. And again, I wanted to take the moment to talk about this a little bit, because I don't think, maybe Yarrick does, but a lot of us don't necessarily conceptualize a lot of this at this level, and I think it's important for us to remember. So with those principles and concepts sort of laid out for us, I'm going to spend the next little bit talking about some elements of the ReproNim toolkit and some applications. So phase 1 and 2 of ReproNim developed all sorts of tools and things. We're not going to talk about all of them. I'm going to try to select a handful of quick success stories, I hope, from the real world in the use of a number of the tools that we have developed. The first one I want to spend a moment talking about is ReproSchema. ReproSchema is a framework for structured survey development and data collection. It provides a library, a user interface, a server backend, and a command line tool for preparing new assessments. Its innovation is that it applies reproducible neuroimaging principles to assessments, allowing behavioral and survey data to be included in workflows and shared more, transparently. ReproSchema provides both the back-end infrastructure and the front end to, immediately use, that back-end technology. And there are some prerequisites and knowledge requisites and technical requisites as listed here. One of the important things here is, again, in the sort of digital side of things where the data is, already digital coming off our MRI scanner, we get lulled into what we can do, electronically, with the content, of imaging data. The much harder domain is the behavioral and clinical and assessment domain, where the information isn't starting digitally. It's starting from a human doing a survey, or a research assistant filling out a form, or things like that. A domain where the data is not as intrinsically electronic and easy to track these kinds of details for. So again, we need the digital interface between the forms and structured surveys and things like that to get this data immediately upon acquisition into as standardized and structured and well behaved of a form, and try to get rid of the Excel files and random CSV things that lie around where a lot of people will often keep data. So that's kind of the background to ReproSchema. Conceptually, ReproSchema takes a structured instrument, a survey, takes the questions and creates a schema for surveys. The items have a particular place in the schema. The answers have a particular place in the schema. The categories all have particular spaces. The responses all have particular spaces in the schema. And each of these therefore can also have sort of additional high level metadata about what the survey is covering, and what the different answers may relate to in terms of behavioral and clinical properties. I don't want to dwell on this. You can read the lovely details in the references. But the point is, generalized surveys have a specific schematized representation, and this, can be shared. This can have the ontological and semantic, content added to it in a way that doesn't get in the way of the survey, but then gives a nice standardized way to share and express survey-based data. This has already, been adopted within the HBCD data organization. HBCD is the Healthy Brain and Child Development Initiative, whose goal, I think, is to scan 10,000 babies or infants from birth to 10 years of old, sort of the child and infant predecessor to the ABCD study, which covers sort of 10 to 20 years old. So within HBCD, ReproSchema has a utility as a schema for defining common structured format for their questionnaires, and it's also provides a platform to store, version, collect, and track the questionnaires that are being collected. So within the HBCD study, researchers using REDCap, that data then gets transferred to a centralized database in Loris, and then the ReproSchema enables the standardized and accurate transformation from REDCap to Loris, ensuring consistency and preserving integrity across the platforms and phases. And this also gives a version-controlled, way to track the status of the evolution of studies. Again, studies are not static. They don't start out and stay the same over time. Studies evolve, questionnaires evolve, and this gives a way to track the evolution of those as well. The next little snippet I want to talk about is, is DataLad. DataLad is a command line tool for data management and sharing. You can download existing DataLad datasets and also assist one in sharing their own data, and can help monitor and track changes to data and supports data versioning. The innovation is by applying source code best practices to data Datalab has been able to build on existing tools to rapidly build a usable system for data management, versioning, and sharing. Requisite knowledge and technical requirements are listed there. Datalet is a good example of, one of the aspirational goals of ReproNim as well, is that not everything that ReproNim does, had to be developed de novo. Uh, DataLad existed before ReproNim, and DataLad will continue to exist, after ReproNim, as are many other, resources. But these are resources that provide critical functions, and so ReproNim, for its hopefully 15-year existence, can adopt these pre-existing technologies and really insert ourselves, our principles, into these existing things, and through interoperating with existing resources, really magnify the reach of the ReproNim technologies. So DataLad is a good example of a resource that we have adopted and play very deeply with, and that will also give us a sustainability, a way for ReproNim things to last beyond ReproNim and to affect users who don't even know about ReproNim because they use these particular tools. And so via the connection with things like DataLad, that helps us buy into other initiatives such as Open Neuro, Brain Life, Dandy, Human Brain Project, other types of things because of DataLad itself's ubiquitous use in these different resources. Oops, wrong way. And thanks to, I think, Adina for generating this. We're not going to go through the details of it, but what I wanted to highlight, a point that I sort of made, is that back in 2016, ReproNim, sort of inserted itself, in the sort of development cycle of the DataLad enterprise, and sort of by our insertion of ourselves into that, at this point in time, that trickles down or trickles out to all sorts of other resources that the DataLad empire is in touch with. So again, there's a lot of utility, a lot of activity, a lot of other projects that use DataLad, and any improvements we can make to DataLad from the reproducibility point of view, trickles into all these other reusers of that technology. And so again, I think this graph in this figure in a way kind of helps show that kind of amplification. Also indicated in this, I think, is the quick application I want to talk about, the fairly big analysis, which is summarized here. That is a framework for computational and reproducible processing of large-scale data. You can read the abstract yourself. Highlights are introducing DataLad to support reproducible processing in compliance with open science mandates, provides a way to capture the machine actionable computational provenance records, gives you the ability to retrace and verify the origins of these research outcomes, and permits sort of re-execution independent of the original computation, and documents sort of the scalability to UK Biobank sized data applications. To summarize super briefly, within the DataLad framework, a process can be a containerized workflow can be run as simple as a DataLad containers run type of command with the necessary command arguments as well. But again, ultimately, containers run is the base operation to take a data set and apply a containerized analysis to it. And now this can be executed once. Take one data set, container run that, you get your result. The importance of the provenance, though, is that because that's all monitored, the ability to do DataLad rerun becomes available, since the run itself captures all the provenance necessary. That permits rerun to be an opportunity. So now each individual subject or analysis that you do, yes, you do it, but it can also be redone, relatively simply. And there's, framework for provenance and metadata, that tracks along with that enables that capability. So while that may be fine for one subject, but so if your data set contains one subject, that's great. But there's no support for large data sets also exists. So instead of a data set containing one subject, a data set could for example contain 42,000 bid structured data sets. And that same basic building block of how to container run, the ability to rerun, and the ability to sort of in a Yoda fashion incorporate the data, incorporate the pipelines, incorporate the results all together in a fashion that can then be distributed across large data sets is facilitated. And so through applications of that sort, the details of this figure are not so important, except that now sort of UK biobank 42,000 subject style observations can be generated. Maybe not quickly and maybe not cheaply, but the framework and the re-executable supports can be built all in through the data lad constructs. Speaking of workflows and workflow managements, the other tool I want to spend a minute on is called NiPoppy. It is a command line tool to manage a complete neuroimaging processing workflow. It organizes and converts raw data, processes it with pipelines, and extracts the derived data for further statistical modeling and analysis. It's the first end-to-end tool integrating other ReproNim tools. NiPoppy addresses the gaps by having identified and to avoid creating ad hoc solutions. NiPoppy creates a roadmap for use of tools and standards for describing workflow processes. Again, sort of covering the workflow environment from data capture through data organization, through data processing, through tracking and extraction of the derived features, the complete set of processes that may happen in the context of a workflow can be captured, and the schematized and represented in a way that supports re-executability and supports distribution of the same workflow to other environments. So this is just a little example of the different elements from the organization, the bidification, the running of processes, the tracking of the executing of the processes, and the extraction of data are all codified in the NiPoppy framework, which then lets you take individual studies and execute them and generate the set of resultant data from individual studies. But wait, individual studies aren't enough. If you take your poppy and then take your bagel, you get a poppy bagel. No, you get a neurobagel. So neurobagel is sort of the next level of that system that lets you do distributed data sharing and discovery, including tools for querying, annotating, and adding your data to a larger federated network. Again, it's the front end to the data sharing technology, immediately usable, and helps support sort of distributed data sharing. So building upon the NiPoppy framework, where an individual site may do their data, and an individual site may can then use the neurobagel to enact the harmonization of the metadata across the different subjects, different studies that an individual site may do, but also lets you share your site to the worldwide collection of these neurobagel representations, so that can be shared either explicitly with collaborators or shared publicly, so that the network of shared resources become, using a common data standard, exposes them to sort of common query that can go across these federated sources. So each site is in control of their own sharing and their own access controls, and can be shared to the world. To date, neurobagel has a huge uptake in resources that are making their data and metadata available. This is just a map that shows a large number of US and Canadian locations, including the Repro Lake that we'll talk about in a minute, and also international uptake of these initiatives, particularly through the various Parkinson's initiatives. But each of those nodes can be searched across to the extent that the searcher is authorized, and each node itself can enhance the ability to search within that node. And that's an example of that is seen in that the Open Neuro interface, public access to Open Neuro, now includes NeuroBagel functionality to do searching within the Open Neuro data sets, and again so within a particular shared resource, and again that content is shared to the federation, so that can be queried amongst with other resources from the NeuroBagel general query platform. So that really brings us up to this next sort of high-level concept of a Repro Lake. Repro Lake is publicly available triple store that contains information on publicly available neuroimaging data sets. The data within the Repro Lake conforms to an IDM standard and can be queried using NeuroBagel or a Sparkle endpoint itself. You can use tools such as PineIDM and others to query that data set. And again, it accomplishes this ability to share a formal structure for public data that preserves the provenance and preserves the annotation of that and the harmonization of those data sets. The Repro Lake content to date includes both the imaging and behavioral metadata from BIDs for the Open Neuro and a number of the indie initiatives, Abide, ADHD 200, Core, and others, and also derived results metadata from tools that have been applied to those above data sets, including FreeSurfer, ANTS, FSL, ANAT, fMRI-Prep, and MRI-QC. So there's a large data store that is represented in that kind of resource that can and is growing and is designed to encourage people to add to that from the publicly available data sets. An example of the use of this is in what we internally refer to as Simple 2. Forget the reason, don't worry about the reason why it's called Simple 2, but it is a use of the Repro Lake that takes a pre-registered set of hypotheses, in this example, about IQ and brain volumes in typically developing children. It supports data discovery for subjects that have the necessary metrics, i.e. IQ, and a volumeable imaging data set, such as found in Abide and ADHD 200. It, in this case, supports the idea of generating a containerized analysis workflow that generates the desired volumetric results from the hypotheses. In this case, using 3 different tools to look at tool differences for volumetric observations, including FreeSurfer, ANTS, and FSL. The results of that get uploaded to the Repro Lake, which can then be queried and then supports the statistical tests of those particular hypotheses. A simple example that Simple 2 looks at, again, is a sequence of brain IQ relationships. One example is from this particular article here that hypothesizes that total brain volume will positively correlate with IQ in both sexes across the complete child and adolescent brain age range. And this figure here on the right is the sort of traditional publication version of that. It's a graph and a PDF, which you cannot really, other than the words that are in that PDF, there's no sort of re-executability or detailed information about that other than what they happen to snapshot into that PDF. The ReproNim version of this, where we can control the way the data is annotated and the way the analyses are run, the results of the Repro Lake query can generate, results for, corpus callosum volume and total brain volume, and generate a similar, correlation. But in this case, every element of the ReproLake content, knows, who the subject is, what the demographics and clinical variables were, what the MRI scan was, and where that scan is, in the shared, world, and what the workflow that generated that particular answer is, what software, what version, what container, and things like that. So, in addition to being able to collect the data, each of the data elements are known and can be regenerated, and the process that generated these can be used on additional data to add to that particular observation. So in this case, the original hypothesis came from an article that didn't quite get a significant no, did. No, so our reanalysis of that generated a fairly significant correlation between brain volume and IQ. The original report was only sort of trend significant. So the point is the original observations can be modernized, can be standardized, can be re-executablized in sort of this living fashion, so that and housed in resources like the ReproLake to support this and other types of questions going forward. So with the last few minutes, spend a little bit of time talking about the future. As I said, we're kind of in the run-up to our phase 3 competing renewal. So a lot of this is fresh on our minds about how we're trying to phrase this. So again, expressing this to the community and getting your feedback is always is helpful to us. The future is you and the future is me. And so therefore, that means everyone. This includes additional continued technology development, and this includes continued engagement with the community. And our engagement with the community is embodied in our training missions, in our collaborative and service projects, and in our sort of dialogues and interactions between the ReproNim team and the rest of the world. We'll talk about these things briefly in the last couple of minutes. So the themes that are probably going to pervade our renewal focuses on incorporating the SIOPS framework. Well, there's a link in these slides to defining SIOPS more, and we'll see a picture of it in a moment. But again, the goal is to achieve higher levels of operational maturity in the adoption of the ReproNim technologies. So we need these to be more automated, more robust, more comprehensive. And so we're trying to move up the scale of SIOPS, and I'll show that in a slide in a moment. We know we need to embrace variability, embrace the multiverse of analytic flexibility. So it's not sufficient to say one tool or one approach can answer any given hypothesis, but we need to enhance our ability to support multitude of ways that can be used to address any particular question, sort of embrace that, especially when there isn't ground truth. The details of analytic variability and sort of methodologic stability really provide important insights to the consensus around any given finding. And finally, in this day and age, you can't do anything anymore without the use of AI and ML technologies. One of our biggest hurdles is in metadata generation, metadata annotation, metadata representation, and turning hypotheses and questions into query. These are types of things that LLMs are probably very well suited for. So we think that AI ML technologies, LLMs, will be a boon to trying to overcome the last barriers for more comprehensive, more easy to use metadata generation and query generation. SIOPS, as I foreshadowed, is this continuum from ad hoc, processing all the way up to, automated workflows, all the way up through sort of AI closed loop discovery types of things. To date, I think we've done a pretty good job of motivating life up to these sort of shareable process. A lot of the data and software systems we work with, are open source. FAIR is being adopted. People can share processes and data and workflows. They don't necessarily do it, but they certainly can. And so there's no reason why someone, can't relatively easily get to this level 3. But what we really need, is to get beyond that, to the automated workflows, to the closed loop discovery. And so that's kind of the SciOps framework where we're trying to push, higher up into the automation and into the discovery, types of elements. In the training aspects of ReproNim, we use this trickle-down training model, and not a pyramid scheme, not a Ponzi scheme, but it's a trickle-down training model. ReproNim is engaged in a number of direct-to-user types of trainings. ReproNim also runs a train the trainer, the INCF ReproNim Fellowship, which helps train people who then can magnify the training effect to additional folks. And then, as we've already talked about, is we work with other developers in order to get other tools to use ReproNim technology so that every user of these other tools become affected in a good way, with the ReproNim influences. So even if we can't get every user, if we can get every user who uses FreeSurfer, or every user who uses Neurodesk or BrainLife or any tool, you name it, if the tool can be affected, then the users can be affected even if they don't know it. And so that's, again, one of the bigger magnifying factors to get to the end users is through collaboration and work with the various developers. There are many tools and resources from our portfolio of collaborative and service projects. So, again, you can find ReproNim influence in all the different resources and more that are listed here. The training network is vast, based on a core ReproNim curriculum around reproducibility basics, FAIR data, data processing, and statistics. That provides the basis upon which things like the ReproNim Fellowship happen, through by which various workshops and symposiums can be deployed, various hackathons, various courses can be developed and are developed using that core curriculum. In addition, a number of application sort of initiatives have spun off from the ReproNim empire, including the ABCD ReproNim R25 that Angie Laird runs, the Repro Rehab initiative that the Lou runs, Neuro Academy, NeuroMatch, and other initiatives that I've probably missed adding here. So again, by taking this core set of things and applying it to other programs, we can get a magnification on the reach of our training network. So I've showed you a bunch of lovely things, and fine, you could listen to this video to learn all these things, but you too could find this information, and we are in the midst of releasing a new and improved website. Much of the approach that I've taken to presenting this today sort of comes motivated by that new website. You can access it at this particular thing, preview. If you do that, you can volunteer for community focus groups, contact us, and we can add you to our list of community contributors to that. If you're that type of person, you can go direct to our repo and file issues there. If you go to our website, what will you find? You will find a new front page, which is better designed to hopefully answering the questions that people want to get to, getting started, the tools, the fellowship, why reproducibility are kind of our initial entry points. You will find revamped information about the about us things, better access to who we are, who our collaborators are, and the types of things we do, and the why, of the things, that we do. So about in many different ways. You'll find enhanced description of our tutorials and advanced descriptions of our tools. Uh, so again, much easier to find standardized and representative and comprehensive information about each of these and much, much more. I'm going to end with some shameless plugs. Shameless plug one is it's very important that while it's fine that I say these things and ReproNim says these things, we really do need to be reacting to what the community needs and wants. So there is a survey. Perhaps all of you have been inundated with information about it, but if you haven't, scan the QR code, let us know, take a picture of that link, go fill it out. It asks a number of questions depending on how much time you have. But again, ultimately, we're trying to get at, if you can solve, one problem and bottleneck, what it is and what you feel the greatest unmet needs are. And then from those, short answers, it delves a little bit more deeply into some of the details of the ReproNim approach. But please, please, please do that survey. We need to know what people think and what people need, because that drives the details of what we can do. And it's important to have the community. It's important that we are responding to what the community needs. Shameless plug two is an upcoming hackathon at the Society of Biological Psychiatry. So kind of parallel to the OHBM hackathons, we're trying to have a similar presence within the biological psychiatry group. It's going to be April 22nd and 23rd. If you're going to biological psychiatry, or if you're not, if you want to go to Toronto in April, by all means, show up. The contact information is here or on the web. Shameless plug three is I mentioned Repro Rehab, but they have a learners and TA program that is taking its applications for its next year. So if you are in the rehabilitation science area and think Repro Rehab as a TA or Repro Rehab as a learner would be good for you, encourage you to follow up with that. Shameless plug four is OHBM things. First of which is a educational course on analytic flexibility, led by our own eBay and colleagues. That will be the Education Day, 24th of June in Brisbane. And as always, ReproNim and others will be participating in the OHBM Hackathon 21 and 23, 3 days before the main meeting. So that brings me to the end of my prepared materials. Thank you for listening. Get involved. You're already involved by watching this or attending this. So you know how to contact us. You know that we have a blog. We have mailing lists. We have training resources. We have collaborative and service projects. We have the fellowship and the webinar series and office hours. Please avail yourself of those as best you can. So I think that is the end of my prepared statements, and I will stop that for a moment and see if there's questions or.
There are a few comments in the chat, actually. So there are just a couple of pointers there's a pointer to the survey, a pointer for an additional link goes to the same thing for the new site. And a comment from Alice Chen, actually, which would be worth it for you to cross-check.
Does Alice want to speak this, or should I read it?
Yeah, I can talk about it. So the NF Core is a software development community, mostly, I guess, like predominantly it's been used for bioinformatics research software, but it looks like they have a new special interest group representing neuroimaging. And I think they have basically the same overarching goals, looking to assess what are the existing guidelines and where the community is going in terms of reproducible data pipelines. And that's their primary goal. And then the secondary goal is the same, educational contents to onboard new talents.
Oh, good. So thank you for bringing that to our attention. And despite my whole sort of sounding kind of ReproNim centric, I am we are aware that this is a large community. A lot of folks are working on reproducibility from many different aspects. And again, I gave you kind of our view of that, but we really do sort of hope to fit into a network of folks with these similar issues and try to see how best we can learn from them and apply, spread our approaches to other sources as well. So I definitely will follow up with those links, and thank you for bringing it up. And I know many people in the audience were actually the originators of things that I talked about. So the opportunity for anyone from any of those resources or things to correct or improve anything I mentioned is, of course, appreciated.
Maybe one comment, Dave. There might be, I don't know, I like the fact that all those tools and things are contributing to reproducibility in research, in neuroimaging. I think that's the kind of integration of ecosystem or use cases aspect of what do you use for what and things. I think it would be critical for like a practical usage of things, like like not everyone will be using NeuroBeagle for making data findable and when is it useful, when it's less. Not everyone will be using data because of sometimes the technical kind of height of the of the tool and maybe for bad reasons, but whatever. I don't know, how do you think and how do you feel about that in that map of tools developed by ReproNim? What are the kind of could we emphasize some of the persona and lay out what kind of a trajectory and use of tools they could get, or just like to emphasize I mean, like a possible integration of those ecosystems or those tools. I don't know, what do you think of it, like I mean, just like pointing maybe to danger of all those things are developed a little bit independently, and that's good, and like that's practical way of developing things as well. But then like sort of the integration or the kind of crosstalks or the like interoperability of things, where do we how do we deal with that?
Yeah, I'm glad you brought that up. One of the other aspects of the website that I didn't bring up is we also do an organization, a cut on some of the organization around some user personas. Those personas, kind of generically are sort of single lab, juniorish people or multi-lab, collaborative, collaboration folks, and sort of teachers, and there's a fourth one, whatever. So there's a number of personas there where we try to highlight different tutorial pathways that go through some of the things. We have written or have initial versions of one of those pathways, but again, I think as you're kind of alluding to, there are many different ways to skin a particular cat, although we like cats, that's okay. But again, collecting in the website or in our materials, sort of multiple different pathways to solve specific problems that do or don't use some of the particular tools, kind of the different solutions, as you mentioned. How do you get from here to there if you don't use DataLad or if you don't use NeuroBagel, et cetera, et cetera. So I think we just have to build up the use cases and build up the examples of those, because we can't, people starting de novo, we can suggest something, but again, there's always factors that affect that. So I don't have a good answer to that, but I think it is important to try to cover that. And Marianne adds, try to make the tutorials focus on achieving an aim.
Yeah.
And the tutorials aren't. Yeah. Go ahead, Marianne.
Since I'm here, I was going to jump off, but then I got involved. Yeah, so we tried. Don't know if we succeeded. That's what we'd like your feedback on. But what we were going for is not having a tutorial on here's how you use repro schema, but here's why you would use repro schema to do a certain thing that you need to achieve, either through the principles or the actions that we laid out. So we try to make it more focused on the task. As JB says, there are multiple ways that you can solve that task. We decided some of them required more skills and more investment than others. So we tried to match that to how, the skill set and the budget of these user personas. Basically, it's like how much time and effort can you spend or are you willing to spend? So again, we don't know that we achieved all of our aims, but it was really designed towards saying, as Dave always emphasizes, there are many different ways that you can do this. We have a set of things to ReproNim. Here's how you would use them to do the aim, but the important thing is the aim, right? That you achieve the aim, however it is that you do it. So would be really interested. We know there's still some gaps and things in the way that the website is organized, but would really appreciate if any of you are willing to take a deeper look or participate in a focus group, and kind of let us know whether this is at all useful. That was sort of the intent. And as I said, we also, for some of these, things are constantly being developed. There's constantly new tools coming in, and we sort of feel if we tell you about every possible tool, it kind of confuses things, right? At the same time, over time, as people become more familiar with this, you want to know what the best tool is and what other tools actually do the same things that may be in a different way or is more matched to your skill set. So I think that is something that we are going to constantly add to the website, right? As things come on, as new ways to do this come on, we're going to add those. But I think the more important thing is that we're trying to give a path, a possible path, and then what it is that you're trying to achieve. And you can either achieve it through this path of tools, or you can use your own tools to do that. And, sort of, we're just at the very beginning of that, but we really would appreciate any feedback that you have.
Thanks, man.
I know we've run over time. Thank you for staying a little bit longer. For the record, I do need to say that April 4th will be the next webinar, and Chris Rorden of Micron and DCM to NIIX fame will be presenting to us. So,
Yeah.
I have a quick question, if there's time.
Yeah.
No, I think you guys have worked on so many things, starting from standards to tools to tutorials and training. I'm curious to know, in your experience, what has worked best in terms of getting people to adopt these things? Is it the tutorials? Is it just in-person trainings? These type of seminars, just meeting somebody in conferences? What is the most effective path to evangelize this cult, I guess?
That's a great question, and I'd be fascinated to hear what the others think about that as well. I ultimately think the persistence at hackathons or persistence at conferences, people run into ReproNim influences, in almost every hackathon you go to, and eventually, even if you try to ignore it, eventually you succumb because you've heard too much. And Yarek does a really cool thing about data lab being so cool, or JB and his team do such a cool example of Neurobagel or NiPoppy does a really cool thing. People see it, people say, oh gee, I want to try it. And when you try it with people nearby, like at the hackathon environment, I think that's the kind of thing that is fairly develop some of the usership. I don't know that people go to the website and suddenly say, oh, I want to try this or not. I think the personal interactions are probably the most effective. I don't know if others agree or disagree with that, but that was the answer I came up with off my cuff.
Might as well a little bit depend on the tools.
Yeah.
Which so yeah, and who do you want to reach? But yeah, I think you're right. persistence is often the answer.
Well, and again, I think your own success story with the PD network, again, is a consortium of people with a dedicated need to do something, and getting your technology in front of them was a good way to get pretty good adoption in that of that,
But it
tool.
worked. It worked because Nikhil and Michelle and others went to Amsterdam and showed the things, and
Did a
that's
hackathon, yeah.
yeah, it worked. I think because of the meetings and the yeah. Uh, so the yeah, I think and yeah, that's so I'm doing some advertisement, but really the key thing that actually worked is people who have developed those things, showing them and demonstrating, and I think that's what has worked best.
Again, emphasizing the importance of our collaborative projects like Enigma and the Enigma consortiums, trying to get through Open Neuro, get through ABCD and HBCD, and sort of their user groups, as sort of audiences who are closer to some of the tasks that we're trying to promote.
Cool. Thanks.
Sure. Okay. Unless there are any burning questions, I guess
Take
we
care.
will call this meeting to an end, and thank you all for joining, and see you in the Reprosphere.